Dendritic cells play a key role in initiating the adaptive immune system. T-lymphocytes are unable to see whole pathogens or antigens and require their complementary antigen to be presented to them via the major histocompatibility complex proteins. Only then can they become activated and mature. Central to this education of T-lymphocytes is the dendritic cell, which samples its local environment and if it detects a threat, travels to the lymph nodes to alert the T cells, initiating the adaptive response. The importance of dendritic cells can be summarized by this quote from Professor Ralph Steinman, who won the Nobel Prize in 2011 for his work on dendritic cells. A properly functioning adaptive immune system signifies the best of life. It is diverse beyond compare, tolerant without fail, and capable of behaving appropriately with a myriad of infections and other challenges. Dendritic cells are required to explain how this remarkable system is energized and directed. And that's what we are going to discuss in this video, the central role of the dendritic cell. Of course, we all know that the adaptive immune response is not always perfect. It is not tolerant without fail, as sometimes it can turn against our own body cells, resulting in autoimmune conditions, or it may miss the challenge posed by a cell that has become cancerous, allowing a tumour to develop. But without the adaptive immune system, we would certainly succumb to illness and infection far more frequently. Dendritic cells, as we have previously discussed, are primarily a tissue-resident cell, where they are constantly sampling the local microenvironment in our tissues and organs for the presence not only of pathogens or antigens, but a commensal microflora and other environmental stimuli such as the food we eat or stress hormones, which can all impact on the immune response. Dendritic cells do this by pushing these dendrite structures between cells, which protrude into the local tissue environment, for example the gut lumen, the skin, or the airway, and you may see this described as periscoping. Dendritic cells play an important role in the initiation of antigen-specific reactivity by activating the naive B and T lymphocytes in the lymph nodes and educating them to initiate an appropriate immune response and drive the formation of immune memory. The key features of the dendritic cells that allow this to happen are their tissue distribution, migration through the lymphatics, maturation, and antigen presentation. In their immature state in the tissues, dendritic cells are highly phagocytic cells, constantly sampling the microenvironment through phagocytosis of solids and macropinocytosis of fluids. If they detect a threat, they will migrate down the afferent lymphatic vessels to a nearby lymph node, where they present the antigen they have engulfed on their surface to naive T lymphocytes to generate an antigen-specific response, which is what the adaptive immune response really is. So here is a summary of that process. And what we are looking at here is the lung as an example. In the lungs, we have immature dendritic cells that are sampling the airway, acting as innate sentinels, but also acting as a link to the adaptive response. If a dendritic cell phagocytoses an antigen, then it will become activated, and several phenotypical changes occur. One of these changes drives the migration of the maturing dendritic cell down the afferent lymphatic vessels to the lymph nodes, where it enters the paracortex and presents the antigen to a naive T lymphocyte, which then undergoes expansion and differentiation to become an effector T cell. This effector T lymphocyte leaves the lymph node via the efferent lymph vessel and enters the systemic circulation, following a chemokine gradient back to the lungs. As obviously, if the threat originated in the lungs, we need the T cell to return to the same tissue to carry out its effector functions. It leaves the blood vessel in the lung capillary bed via diapedesis, which we discussed in more detail when we looked at inflammation in general, and then migrates towards the pathogen to clear it. And during this process, it develops immune memory. The dendritic cell is covered in receptors, ranging from pattern recognition receptors on the plasma and endosomal membranes and danger receptors, allowing the cell to respond to pathogen and damage-associated molecular patterns, or those PAMPs and DAMPs. It also has a host of co-stimulatory molecules, which are required to activate T cells by producing additional signals, which act as checkpoints to ensure cells only become activated when required and in an appropriate manner. There are cytokine and chemokine receptors so that the dendritic cell can respond to chemical stimuli, and we will talk about the importance of one receptor in particular later in this video. There are solute transporters for things like glucose, so that the cell can fuel all the activity it has to undertake and produce sufficient levels of ATP. There are, of course, MHC class I, which is on all nucleated cells. But being a professional antigen-presenting cell, it also expresses MHC class II to activate both CD8 positive and CD4 positive T lymphocytes directly. Just as an aside, an easy way to remember which class of MHC interacts with which T cell, the combination of multiplying the class number by the CD number always results in 8. So CD8 multiplied by MHC class I equals 8, and CD4 multiplied by MHC class II equals 8. Back to the receptors, and there are plenty of adhesion molecules, such as intracellular adhesion molecule 1 or ICAM1, and these are used to stabilize the binding between the dendritic cell and the T cell during activation. Finally, the catch-all other receptors, for example sensors of cellular stress or hormones and vitamins, allowing the cell to further integrate information from the microenvironment to tailor the immune response. So now what we are going to focus on is what happens after a dendritic cell takes up an antigen and the process that drives it to the lymph node. When a dendritic cell is in the tissue and it hasn't encountered an antigen, it is in an immature state. It has low expression levels of costimulatory molecules, low levels of MHC class II. It does, of course, also express MHC class I, but we tend to focus on MHC class II when talking about dendritic cell activation. It is not secreting any pro-inflammatory cytokines, but it is an exceptionally good phagocyte, constantly sampling the microenvironment. It has low expression of the chemokine receptor, CCR7, and as it's just ticking over in the tissue sitting there, it doesn't need much ATP, so glycolysis is low. Upon activation by a pathogen, by cytokines, or if it detects PAMPs or DAMs, then the dendritic cell undergoes a host of changes, upregulating its expression of costimulatory molecules and MHC ready to activate T cells. It starts secreting pro-inflammatory cytokines to switch the adaptive immune response on. Its phagocytic capacity drops as it's done that part of its job and is now moving on to the next stage. CCR7 expression on the surface increases, and this allows the cell to respond to a couple of key chemokines that are released from the lymph nodes, and it guides its migration through the lymphatic vessels. And because the cell is now much more metabolically active because of all these changes, it enhances its glycolytic capacity. We mentioned the upregulation of the chemokine receptor CCR7 as part of this maturation process. And this receptor allows the dendritic cell to respond to two key chemokines that are produced by the lymph organs, called CCL19 and CCL21. As well as causing the maturation of the cell, these chemokines drive its chemotaxis from the tissue into the lymph nodes, affecting its migratory speed, changing the cytoskeleton composition, and enhancing endocytosis capabilities. The reason that the upregulation of this receptor is important is because it allows the activated dendritic cell to respond to these key chemokines. So although these chemokines were present in abundance in the lymphatics, the immature dendritic cell, which had low levels of CCR7, were unable to respond to them. Now both CCL19 and CCL21 bind to the same CCR7 receptor, but to slightly different amino acids. And we previously introduced the concepts of pleiotropy and redundancy in cytokines and chemokines. The CCL19 and CCL21 have different effects, with the thickness of the arrows giving an indication of the contribution of each chemokine. The CCL19 has a greater effect on chemotaxis than CCL21, but CCL21 enhances the phosphorylation of the ERK signaling pathway. Hence, both chemokines are needed for optimal dendritic cell activation. What happens once the mature dendritic cell reaches the lymph node? Well, we mentioned that the dendritic cell had a large number of receptors on its surface, and a key aspect of the dendritic cell's role when it reaches the paracortex or the T cell zone of the lymph node is to bind to naive T cells and present the antigen that is engulfed. In order to activate T cells, the dendritic cell needs to provide three signals, which are called signal 1, signal 2, and signal 3. Not very imaginative, but at least easy to remember. And these signals occur in order. So signal 1 comes first and is the interaction between the MHC presenting its antigen in the peptide binding groove to the T cell receptor. Signal 2 comes second from the costimulatory receptors. The example here is CD28 on the T-lymphocyte binding to CD80, but there are many more and we will look at a few later in the video. And finally, signal 3 comes from cytokines released by the dendritic cell that acts on the naive T cells and elicits a response that is appropriate for the antigen that initiated the process. So, if it is a virus that has initiated all these processes, we want to ensure that we mount an effective antiviral response to combat that virus. And the extent and type of these three signals is determined by the signals that the dendritic cell is integrating from its environment. We've discussed the major histocompatibility complex quite a lot so far. Let's take a closer look at this. MHC and HLA, or human leukocyte antigen, tend to be used interchangeably in humans, and this really reflects their discovery, with MHC being found in mice about the same time as HLA was being described in humans, but they are essentially the same thing. MHC, or HLA, is encoded on the short arm of chromosome 6, and is the most polymorphic region in the human genome. It actually encodes three classes of HLA, but for the purpose of antigen presentation, and therefore simplicity, we only need to look at class 1 and class 2. Within class 1, we have three different types of class 1 HLA molecules: HLA-A, HLA-B, and HLA-C, with several thousand alleles within each subfamily. For class 2, we encode HLA-DP, HLA-DQ, and HLA-DR, and again, there are thousands of different alleles in each subgroup. Our MHC is inherited in blocks of alleles known as haplotypes, and we get one set from each of our biological parents, as they are co-dominantly expressed. Everyone has 6 MHC class 1 alleles, one each of A, B, and C from each parent, and we get between 6 and 8 MHC class 2 alleles. We inherit one each of DP and DQ, and either 1 or 2 copies of DR from our parents. If we look at this family tree, we can see the different inheritance patterns, with each offspring inheriting a slightly different haplotype, receiving half each from their mother and father. And this would be the type of information that would start the matching process if one of the children needed an organ donation, to identify which of the siblings might be a good potential donor, although the process does go a lot deeper than this. We've seen the structure of the MHC class 1 and class 2 molecules before, but just to remind you that the class 1, which is expressed on all nucleated cells, is made up of a series of 3 alpha domains and an associated beta-2 microglobulin domain, with the peptide binding cleft formed by the alpha 1 and alpha 2 domains. MHC class 2 is only expressed on antigen-presenting cells and is made up of an alpha chain and a beta chain, with the peptide binding cleft made up of the alpha 1 and beta 1 subunits. We have a whole host of different MHC molecules, but an important thing to note is that it is not just one MHC molecule per peptide, as that would be rather limiting. So each MHC molecule can bind and present many different antigen fragments. So any given MHC can bind numerous different peptides, and some peptides can be presented in several different MHC molecules. So the combination of all these factors ensures that humans can present a vast array of antigens. Dendritic cells express both MHC class 1 and MHC class 2. The MHC class 1 presents endogenous peptides, so peptides that are derived naturally within our cells, but also any pathogen that invades intracellularly as part of its life cycle. So a virus is a classical example of that, and viral peptides can also be expressed in the MHC class 1 binding cleft. MHC class 1 presents its peptides to CD8 positive T cells, that we call CD8 T lymphocytes MHC class 1 restricted, as they can only see their cognate peptide in the context of MHC class 1. In contrast, MHC class II expresses exogenous peptides, so those that are found outside our body cells and have been phagocytosed or macropenocytosed, so bacterial peptides would be a classical example here. And the MHC class II molecules are only expressed on a specialized subset of leukocytes called antigen-presenting cells. MHC class II interacts with the T cell receptor on CD4 positive T cells, so we call CD4 T lymphocytes MHC class II restricted. So it is important to remember these distinctions. CD8 positive T lymphocytes are class I restricted, interacting with HLA-A, B, and C, while CD4 positive T lymphocytes are class II restricted, interacting with HLA-DP, DQ, and DR. We will talk about the specificity that the T cell receptor, the other half of this interaction, provides when we look at T lymphocytes in more detail. So how does a peptide get into the binding cleft in MHC class I and class II? The processes are slightly different. So on the left here we have the MHC class I pathway, which remember is for endogenous proteins or intracellular pathogens like viruses. Here the protein in the cytosol is broken down by the proteasome, and those peptides then associate with the MHC class I molecule in the endoplasmic reticulum. The MHC class I molecule is packaged into a vesicle and transported to the cell surface where it's expressed. In contrast, the MHC class II pathway on the right, which is for exogenous peptides or extracellular pathogens, requires the extracellular protein to be taken up into an endosome, either through phagocytosis of solids or macropenocytosis of fluids. The peptide is broken down within the endosome and then fuses with a vesicle from the endoplasmic reticulum containing the MHC class II molecule, allowing the peptide to displace an invariant chain that normally blocks the binding cleft, and then the MHC class II molecule is transported to the cell surface for expression. So, that is an overview of the major histocompatibility complex that initiates signal 1. But what about the other signals? Well, signal 2 comes from the co-stimulatory molecules and their receptors. The example here is CD80 on the dendritic cell binding to CD28 expressed on the surface of the T cell to promote T cell survival. We now know that there are both co-stimulatory and co-inhibitory receptors that determine the functional outcome of the MHC-TCR interaction. And there is a diverse range of receptors in terms of expression and function, which control T cell activation, differentiation, and function. As our knowledge of immunology has increased, we have discovered a wider range of co-stimulatory and inhibitory molecules, and we have started to appreciate the complex interplay that happens to shape the immune response. And this is just a small selection, it's not exhaustive. We aren't going to look at every one of these interactions, but to highlight a couple of important ones. Firstly, CD80 and CD86, which may also be shown as B7-1 and B7-2 respectively. And these are expressed on the dendritic cell. And this is an interesting one, because we have shown already that they can bind to CD28 to provide an activation signal to the T lymphocyte, and they are critical for T cell activation. But they can also interact with CTLA4, which provides the opposite inhibitory effect. So the pattern of relative expression on the T cells can determine its fate. What happens during co-stimulation? Well, we see a classical biochemical protein-protein interaction, which results in a downstream signaling cascade. With CD28, there are 3 main pathways that can be activated, depending on what other signals are received, and these result in the upregulation of one of 3 transcription factors, NF-AT, AP1, or NF-kappa B. The important outcomes of these, however, are to upregulate the T cell activity, so it causes them to proliferate and release cytokines, upregulate metabolic processes to fuel these activities, and enhances their survival. The other interaction we're going to look at is PDL1 and PDL2, which binds to PD1 expressed on the T lymphocytes. And this is an example of a co-inhibitory signal, and it's what we call an immune checkpoint, which is very important in the context of cancer. Under normal conditions, they act to ensure an appropriate immune response and down-regulate activation to regulate that response. However, in cancer, these signals can be switched on and inhibit the immune system from acting against the tumour cells. So here we have a T cell receiving signal 1 and signal 2 from the MHC TCR complex, and CD28 binding either through CD80 or CD86. And this would normally result in the downstream effects and the associated effector functions that we have already discussed. However, the T cell can also express PD1, which when bound to either PDL1 or PDL2, results in activation of a different intracellular signaling cascade and inhibition of these processes, as indicated by the red block-ended arrow here. There is a great deal of interest in blocking this PD1 interaction using antibodies or small molecules in the context of cancer to allow the T cells to activate against tumour cells, which can express high levels of PDL1 and PDL2 on their surface to deactivate the T lymphocytes. The area of contact between a dendritic cell and a T cell is tiny, and we have come to appreciate that it is a lot more complex than just a simple protein-protein interaction, as there are numerous proteins and receptors involved, providing activation and inhibition signals, so that's signal 1 and signal 2, but also adhesion molecules that help stabilize the binding. Because we now know that the interaction between a dendritic cell and a T lymphocyte is long-lasting, often over a period of 24 hours or so, with the cells engaging and disengaging multiple times during that time. The resultant area of contact is known as an immunological synapse, and it's highly structured. And this is just a very simplified overview, as there are many, many more potential interactions occurring. And finally, on to signal 3. This is the cytokines that are released by the antigen-presenting cell, acting on the T cell to educate that cell as to the optimal response based on the eliciting agent. The released cytokines can equally act in an autocrine manner on the APC cell or in an endocrine manner spreading throughout the body, but here it's a paracrine action on the neighbouring T cell that we are interested in. The cytokines that are produced by the antigen-presenting cell are as a result of all of the signals that is integrated from the tissue. And while signal 1 and signal 2 really drive the activation and proliferation of the naive T cell, the cytokines will push the T cell towards its final phenotype. So it's already committed to being a CD4 or a CD8 T lymphocyte, but there are further subsets, and this is where these cytokines come into play. So a naive CD4 T cell, depending on the signal it receives, may differentiate into a follicular T helper cell, which secretes IL-21. It can become a Th1 or a Th2 cell, secreting interferon gamma or IL-4 and IL-13 respectively. It may become a Th17 cell producing IL-17, or a regulatory T cell secreting IL-10 and TGF-beta. And we will discuss these different subsets in more detail when we look at T cells in a future video. The outcome of all of this is that the eliciting agent has shaped the dendritic cell response, and it has determined how much MHC class 1 and class 2 are expressed, and which antigens are presented in the binding cleft due to the breakdown of the proteins. It has determined the abundance and type of co-stimulatory receptors and the type of cytokines that are released. As an example, if the eliciting agent was a parasitic worm, in this case a lower parasite, then the downstream effect would be to drive the naive CD4 positive T cells towards a Th2 phenotype, which produces interleukin 4 and interleukin 13, which have an important immunological effect in activating our eosinophils and producing antibodies that can combat the parasite. Conversely, if the initiating threat is a fungus, then the naive CD4 positive cells might polarize towards the Th17 phenotype, which releases IL-17 and IL-21 that can help activate neutrophils and macrophages to defend us against the fungal infection. So that covers the main functions of dendritic cells, acting as a link between the innate and the adaptive immune system. We've looked at how the three signals the naive T cell receives from the dendritic cell can activate the cells and drive the formation of an effective immune response that is targeted and appropriate for the eliciting agent. In the next video, we will look in more depth at T lymphocytes and their crucial role in adaptive immunity.